Good afternoon, everyone. Today we are standing in the field of IRI Pusa campus. This is basically Brassica junctia. Here you can see, which is a cultivated species of mustard. Here you can have a look. Besides cultivated varieties, we have also grown many wild species of Brassica. Here you can have a look. This is crambe. You can have a close look on the flowers. Here you can see another different wild species of Brassica. This is basically Sinapis alba. This is Lepidium sativum. Here you can have a look. This is Ericastrum. This is Diplotexis ericoides. This is Erica sativa. You can have a close look on the flowers. So in this video we are going to demonstrate you how to do the crossing in case of Brassica. Here you can have a look on the Brassica cultivated species. The left one, this is basically Brassica junctia, and on the right side this is a wild species of Brassica, which is Erica sativa. So you can have a look how different they are. So next we are going to explain the different parts of the Brassica flower. So here you can have a look on the flower. This is basically the sepal. There are 4 sepals in the Brassica flower. My colleague is removing them to also show you at the same time how to dissect the flower. Now these are the petals. There are also 4 petals in the Brassica flower. 2, 3 and 4. So we have removed all the 4 petals also. Then you can have a look. There are 6 stamens. Out of those 6 stamens, 2 are small and 4 are big. Here you can have a look. This is the anther, and the attaching portion is the filament. This is the exposed stigma. You can see stigma and then style, and below the style there is ovary. So this is the exposed ovary with all the flowering parts are removed. So let's start with the crossing demonstration. First of all, we have to remove the mature flowers that are present. And also the unmature buds. Here you can see we are removing the. So finally you can see over here we have selected the appropriate buds, just 6 to 7 buds for crossing purpose. So we are going to show you the demonstration how to emasculate these buds. We have to put a cut in the bud. Then slowly we have to remove the anthers or the stamens. All the 6 stamens we have to remove. So these are the emasculated buds. Just 5 to 6 appropriate buds were selected and all of them were emasculated. Then we have to cover the buds with the help of a butter paper to prevent outcrossing. Put a stapler. So the next day morning we have to start with the pollination process. We have to take the pollen grains from the freshly opened bud. We can collect the pollen in a petri dish. So here we have collected the buds from the wild species of Brassica, that is Erica sativa. We are going to pollinate it on the cultivated species, that is Brassica junctia. So this was the selected, ये क्या था? Selected emasculated.
हाँ, this is the emasculated birds.
Bud है ना? Emasculated bud.
हाँ.
और the female parent.
Female parent.
करूँ?
हाँ.
So next day we have to remove the butter paper from the emasculated bud. So this was the emasculated buds. Now this was the collected stamen. This was the wild species that which we have shown, that is Erica sativa. Now enter we have from this flower we have to pollinate on the female parent. So now we have cross-pollinated the pollens from the wild species to this cultivated Brassica species, and then we have to cover with the help of butter paper. After the pollination is over, we have to prepare a tag which will mention the name of the parents, date of emasculation, date of pollination, and we have to tag on that particular branch in which pollination was carried out. So with this, our crossing steps are over. So we thank you all for watching our video.